everybody. It's my pleasure to be with you today. And in the next 20 minutes, I'm going to provide with an overview on Parkinson's disease genome-wide association studies. I'm going to highlight what we have learned so far, as well as the challenges and the limitations. And I'm going to briefly discuss as well key GWAS advances that have been done in the past, as well as what the present and what the promising future in our field holds. So let's get started. So I thought that the best way to start this talk was briefly refreshing the concept of GWAS. So a GWAS stands for Genome-Wide Association Studies, and this is a really popular method, right, that is used to test common genetic variants across any platform that is usually genotyping imputed data. So a GWAS aims to identify possible association, right, between genotype and between phenotype, which in this case is Parkinson's disease. So we say that if a certain genetic variant is found to be in cases significantly more often than in controls, then it's associated with the disease. And you see here, and this is what we do when we want to perform a GWAS, is we compare the differences that exist in terms of magnitude of effects or in terms of frequencies for certain genetic variants at a genome-wide level in our cases and our controls under study. And since we are testing thousands of variants, right, because we are doing this at a genome-wide level, our significant threshold is usually really stringent, since we take into account 5 times 10 to the minus 8. And if we think about Parkinson's disease, we realize that really the majority of the cases do not have clear modes of inheritance, right? In fact, over 90% of the cases are considered sporadic, in which, there is an interplay that we know that exists between those really small effect variants that are common in the general population and then environmental factors. So GWAS is really this right technology to investigate this type of genetic variation. And so, although GWAS is this powerful approach that we use, that is used to give insights on Parkinson's disease etiology, and we do this in an unbiased manner, right? There are many things that GWAS studies do not really tell us. And therefore, there are many questions, I will say, that remain unsolved and that certainly warranty further study. I have included here some of them. So one of the main questions is that we do not know which are the causal variants, right? GWAS identify loci, so identify regions of the genome, but not really causal variants. Then another question is, are SNPs enriched in certain tissues, or do the associated variants that we find linked to disease modulate gene expression? Other questions that we need to further investigate after GWAS is, do we predict risk of disease? Can we do that? Can we create polygenic models of disease? And importantly, how do we translate GWAS loci? How do we translate this information to a functional understanding of disease, right? And of course, penetrance. Penetrance is something that we cannot really find out from GWAS. So all of these questions require further study beyond what GWAS can tell us. But I'm here to be realistic, and in fact, the Parkinson's disease genetics field is very aware of our challenges and our limitations. So in fact, GWAS only looks at common variants, so generally variants that have a minor allele frequency above 1% in the general population. Of course, depending on the sample size on the power, that threshold can vary a little bit. And sometimes we consider a minor allele frequency above 5%. So GWAS is certainly not the best technology to detect rare variants, and generally cannot provide information on this type of genetic variation. Also, GWAS only nominates loci, so those regions of the genome and the nearest gene's protein coding gene, which functionally, really this might not be the causal gene. And then, of course, population substructure is a major issue. So I would say the frequencies, the directionality of effect, the effect sizes that all can be different across population. And the literature shows that loci that are nominated in one population might not necessarily translate to other populations, as we will see later on in the presentation. And of course, quality control and harmonization are really priority to avoid artifacts. And GWAS explained that really a small number of effect associations, which, as I said, functionally can be, I would say, a tough problem to tackle, right? And additionally, of course, because we are screening at a large scale manner, there is a massive multiple testing correction penalty, and therefore we need really large sample sizes to get some insights into the disease etiology. So, however, also I would say, despite those challenges and limitations that I highlighted, in many ways, GWAS studies have been the engine that has pushed us along our voyage to understand Parkinson's disease. And historically, in the pre-GWAS era, there were only very few familiar sequencing and small scale genotyping studies performed. Those were the only possible studies. But now, thankfully, technology allows for cheap, high throughput genotyping of thousands and thousands of samples. And thankfully as well, reference panels for imputation are becoming more and more accurate. So before I dig into the current scenario, I would like to briefly discuss the starting point of the GWAS era in Parkinson's disease genetics. And these two references that I highlight here were the first two GWAS studies that were published in our field in 2009. So the first one that you see by Satake et al. In the Asian population nominated SNCA that encodes for synuclein and BST1. And then we have the second one here by Simon Sanchez et al. That uncovered, again, synuclein and MAP in the European population. And this was the first time that we could see that the pleomorphic nature, right, of these loci, in the way that, for example, point mutations and duplications and triplications in synuclein encoding for, in SNCA encoding for synuclein, are linked to familiar Parkinson's disease, as it was previously presented in the last presentation, with a large impact on disease etiology. But then we have, on the other end, genetic variants in SNCA as well that are common and that have a smaller, overall effect on effect sizes on disease etiology, and those are going to increase only slightly the risk for disease. And so from that point on, really great advances were made in identifying many of these loci that confer risk for Parkinson's disease. And this era really led to increased collaboration that allowed the identification of new and important insights on disease etiology. This slide really illustrates how the number of cases increased with time as more groups and more samples were collected. And it should be pointed out here as well that these collaborative groups form the basis of the International Parkinson's Disease Genetics Consortium that has been crucial to promote open science and to connect researchers and clinicians worldwide. So for you to have an idea, this is the first Manhattan plot that is representative of the first genome-wide association study. And it's beautiful, but it's quite empty. And this one that we see here, this is the last Manhattan plot, 10 years later, right? And we see that it's really, really crowded. So looking at where we are now, what have we learned? So the last genome-wide association study meta-analysis that was conducted in the European population, the one that I just showed, the Manhattan plot, allowed the identification of up to 90 independent genome-wide significant risk signals across a total of 78 genomic regions. And this included about 57,000 cases and proxy cases. Proxy cases are those cases defined as individuals that are not affected with Parkinson's disease, but have a first relative that is affected with the disease. And this against 1.4 million controls. So if you see, in 10 years, we went from 2,000 samples to over 1.4 million samples. And this is, I think, really fascinating. And one of the curiosities as well to point out is what I show in this slide. So here you can see two studies, right? We have above the last GWAS meta-analysis that I just mentioned, and then we have below a follow-up epidemiological study of Parkinson's disease in twins, 20 years after the original study. And in this second study, the authors had followed the twins for a further 20 years, right? And some of the cases who were, discordant in the original study were not discordant in the latest study. And in those 20 years, they had developed disease. And it seems that the pairwise concordance for Parkinson's disease in the twin studies, as compared to the estimates of heritability from the latest GWAS meta-analysis, are very similar. And this points to around 27% of heritability, which means the percentage of the phenotype that is attributed to genetic influence. And I think this is a beautiful example of really the power of GWAS studies, because this shows that there is concurrence between that heritability of Parkinson's disease, both from molecular genetics, so the last GWAS meta-analysis, and also from epidemiological data. And in the Parkinson's disease genetics field, current identified GWAS signals explain about 1/3, more or less, of the total heritability of disease. And this means that, of course, there is a lot more to be uncovered, right? And we usually refer this to as missing heritability. And what has come out of GWAS? Well, I will say plenty of valuable information, mostly conducting post-GWAS analysis on this data. And we have learned that there is up to 6-fold increase in Parkinson's disease risk in those individuals carrying the highest burden of risk, including the 90 risk variants from the last meta-analysis. But also GWAS have been, I will say, key at informing biological pathways that are involved in disease etiology, such as, for instance, the lysosome, mitochondrial genes, immune system, dopamine metabolism, etc. We also know, of course, that there are certain GWAS variants that are enriched in specific tissues or cell types. And we have also learned that there are definitely overlapping genetic etiologies and also causality with certain traits and much more. However, we not only know about the genetic architecture of Parkinson's disease risk. I think as a field, we have also made progress at understanding Parkinson's disease progression. And this is the first study reporting genome-wide significance for Parkinson's disease GWAS loci for age at onset, including about 28,000 cases. And this GWAS discovered several Parkinson's disease loci that are influencing age at onset, such as SENCA, again, encoding synuclein, ApoE, and also TMEM175 locus, as well as other subsignificant loci such as GBA, SCARP2, and BAC3. And we also have this other recent and interesting GWAS on Parkinson's disease progression that included clinical data for over 3,000 participants, showing that the ApoE4 allele drives progressive cognitive impairment in Parkinson's disease. Or also, we have as well this other recent GWAS on Parkinson's disease progression that follow up over 4,000 patients, and this one identified 2 novel variants associated with different progression phenotypes. So one variant in SLC44A1 associated with faster development of motor symptoms, and we have also another variant that is a QTL of ADRA2A, and this one is associated with a lower prevalence of insomnia at baseline. And I would like to briefly mention as well and summarize this list of genes whose variants have been also reported to be associated with Parkinson's disease progression in previous association studies. And the most studied genes is obviously GBA, right? And in particular, the E326K variant that has been linked to faster cognitive decline. Parkinson's disease patients with GBA variants seem more affected as well by REM sleep behavior disorder. And then regarding cognitive function, the ApoE4 allele was also reported to be associated with cognitive function in the Parkinson's disease population. And as mentioned earlier, age at onset has been reported to be associated with the Parkinson's disease risk variance in TMEM 175 and synuclein regions, which really correlates the genetic architecture of risk and age at onset. And so after all, what is next, right? So, well, this slide illustrates how, the number of cases has tremendously increased with time as more groups and more samples have been collected, and how important collaboration is to make scientific progress, right? And this is, as you see in this image, this is an estimation of the projection of number of GWAS loci with the increase in Parkinson's disease cases. And what really this image tells us here is that teamwork is crucial, right? Teamwork is crucial to unmask the contribution of currently uncaptured genetic variation to the risk of Parkinson's disease. And importantly, this is where the future goes. And so one of the things that we have to correct, and thankfully it is already happening, is that nearly all the GWAS studies conducted so far have been done in Europeans. And obviously that's not satisfactory, right? And this clearly has to be, a major goal for the future. And I would like to say, and this is something that you will hear in the next presentation, the Global Parkinson's Disease Genetics Program, that is also called GP2, is going to be key to achieve it. And we need to really understand the genetic architecture of Parkinson's disease in more Asians, in Latino populations, and Africa, and so on. And here you see the two first GWAS studies. You see these Manhattan plots from underrepresented populations that have been just published. And as a note, the Asian GWAS, Fo et al., has nominated 2 novel loci that were not previously identified in Europeans, which really tell us that, again, genetics is not universal, and definitely we need to translate this to other populations. And so the future is also in multimodal analysis. And it seems that combining different types of data with genetics is going to lead to much more accurate prediction and, of course, better models. Data harmonization and also intelligent storage of data as cohorts are going to continue to grow in size and in scope. And of course, standardization of these processes, for fast processing of all that data and automatization of this analysis seem to be the way to go. So just to sum up, here is some take-home messages. So as a field, we have certainly come a long way, and however, there is some points for you to remember. So let's keep in mind that GWAS identify common small effect variants that are associated with disease. So these are proxies, right? And these nominate loci, again, regions of the genome that can certainly inform biological investigation, but is not nominating any causal variant. Then by combining genetic information with other types of data through multimodal analysis, we can certainly predict a risk, better predict risk. And biobanks are the present, but are also the future. I think biobanks are going to facilitate findings, because success in the new era of Parkinson's disease genetics is going to require extensive scientific collaboration to get to that sample size. And this is impossible. We'll not be able to make any progress without teamwork in an open scientific environment. And finally, it should be also pointed out that therapeutic targets with with a valid and proven genetic basis really lead to better drug development candidates. And so for those that are interested in learning about GWAS in Parkinson's disease, both from a theoretical but also from a practical perspective, please take a look at our freely available resources developed as part of the Global Parkinson's Disease Genetics Program, also called GP2. So we have the GP2 Beginner Bioinformatics for Parkinson's disease genetics. And then we also have the GP2 Parkinson's disease genetics for non-geneticists course. And the link to register for these courses is here at the bottom of the slide. So with that being said, thank you very much for listening. I hope this was entertaining, and I'll be happy to discuss later on. Thank you.